Lipids are a major class of biological molecules that are important in energy metabolism, cell membrane structure, and cell signaling. Lipids can typically be recognized by their nonpolar hydrocarbon structure. This makes most lipids largely hydrophobic and not very soluble in water. We will focus our lesson on three different categories of lipids: the fats, the phospholipids, and sterols. Though their structures and compositions can be diverse, they share a common biosynthetic origin as polymers of the small subunit acetate. We will present and review the vocabulary, properties, structures, and principles related to each of these important lipid groups. Fats are an important energy storage molecule. It is likely that our palates are designed to recognize and crave fats because they are such a rich source of energy. And fats can be an important part of your diet, particularly if you are a hunter-gatherer and you never know where your next meal is coming from. In this tutorial, we will present the chemical structure of fats, describe their nomenclature and numbering, define the term saturation, and finally, describe how the structure of fatty acids determines an important property of fats called fluidity. A fat is made from a glycerol backbone and 3 fatty acid chains. Glycerol is a 3-carbon chain with 3 hydroxyl groups. These hydroxyl groups form sites for the condensation reactions needed to form a fat. Fatty acids are long hydrocarbon chains, called alkyl groups, that have a carboxyl group attached to one end. This chain has 12 carbons, but the length can vary for different fatty acids. The carboxyl group is always in the same position at one end of the chain. Attaching each fatty acid to the glycerol backbone involves a condensation reaction. In the reaction, a carboxyl group from the fatty acid is brought together with a hydroxyl group from glycerol. A new bond is formed, and a molecule of water is released. The resulting bond is called an ester linkage. The attached fatty acids are now referred to as acyl groups. Attach 2 more acyl groups, and it's easy to see why the chemical name for fats is triacylglycerides. Despite their simple structure, there's a surprising diversity in fats that results from the different types of fatty acids that can be added to the glycerol backbone. For example, fatty acids come in a variety of different chain lengths that vary between 12 and 24 carbons long. A few of the more common chains are shown here, with both their common names and chemical names. Common names, shown in red, are often derived from prominent sources. For example, palmitic acid is found in palm oils. Stearic acid is derived from the Greek word for tallow or lard. Chemical names simply describe the number of carbons in each chain, as you can see in orange. For example, lauric acid has the chemical name of dodecanoic acid. Dodecanoic is Latin for 12. Each structure is shown in the weak acid form if you look at the carboxylic acid group on the left-hand side. However, they can also be drawn as the conjugate base. And that has a corresponding change in the name. So lauric acid, for example, when you draw it as the conjugate base, becomes laurate, or dodecanoate if you're using the chemical name. While previous structures have been drawn as straight lines, you will often see the hydrocarbon regions drawn in a sawtooth pattern to better represent the tetrahedral arrangement of the carbon chain. You may also see an even more abbreviated structure that removes all the hydrogens and hides the carbon atoms at each vertex. Note that in nature, almost all fatty acids have an even number of carbons, reflecting their synthesis from the 2-carbon acetate building block. In some cases, fatty acids contain 1 or more double bonds along the chain. These chains are said to be unsaturated because 2 hydrogen atoms are removed from the chain, leaving it less than saturated. The remaining electrons join together to form the new double bond. Polyunsaturated fatty acids have more than 1 double bond, And fatty acids with no double bonds are said to be saturated because they have the maximum number of allowed hydrogens. So remember, unsaturated simply means that there are one or more double bonds in a chain, and saturated just means that there are no double bonds in the chain. Double bonds in a carbon chain are identified by the carbon number in the chain. Unfortunately, there are two commonly used methods of numbering and naming double bonds. Chemists prefer to start numbering at the carboxyl group and count up to the end of the chain. They use a Greek letter delta to represent this system. Nutritionists prefer to start numbering the chain from the opposite end. They use the Greek symbol omega because it starts at the end of the chain. Omega is the last letter or end letter of the Greek alphabet. Though a double bond involves two carbons, only the lower carbon number is used in identifying it. As a result, this molecule would be known as delta 14 or an omega-6 fatty acid. In recent years, omega-3 and omega-6 fatty acids have received a lot of attention for their health benefits. The omega nomenclature was used because the reports were made by nutritionists. If chemists had made the initial reports, we would be using the delta nomenclature. Let's try a practice problem to see if you have the numbering concept down. The fatty acid shown here is oleic acid, a common component of olive oil. Name the double bond position according to both chemists and nutritionists. We'll give you some time to calculate it. Counting from the carboxyl end, we see that the double bond starts on C9, making this a delta 9 fatty acid. Counting from the methyl end, the double bond starts on C9, making it an omega 9 fatty acid also. This doesn't really happen very often that they both start on C9, but it did in this example. The variation in fatty acids allows us to create fats with different properties and characteristics. One such property is fluidity. Fluidity indicates how well a substance flows from one form to another. Some fats, like butter and lard, form solids at room temperature because the individual fat molecules make lots of weak bond interactions with each other that hold them closely together and limit their ability to move or flow. Other fats, like olive oil and canola oils, form liquids at room temperature. The fluidity of fats affect their taste and their texture. They also affect their metabolism and the health effects in the body. Thus, it's useful to examine the principles and conditions that govern fluidity in fats. Solids form when two or more molecules are able to bond with each other. Two fats associate and bond along the length of their carbon chains through what chemists call an induced dipole interaction. Biologists simply refer to it as van der Waals interactions. Van der Waals interactions occur from short-lasting opposite charges that we'll explain in a moment. Any single van der Waals bond forms or breaks easily, but as one breaks, there are other bonds forming that help the two chains stick together. Watch carefully at the dotted lines holding these two fatty acids together to see what I mean. Individually, they are weak, but collectively they add up. To learn more about the charges that create van der Waals interactions, let's zoom in on a single pair of carbon atoms on opposite chains. The electrons making up the hydrocarbon bonds are equally shared, but constantly in motion. Because the carbons are so close to each other, the electrons try to synchronize their movements so that they don't occupy adjacent hydrogen atoms at the same time. As a result, the electrons of one bond are near the carbon, while the electrons in the other bond are near the hydrogen. This creates a split-second partial positive charge on one hydrogen and a split-second partial negative charge on the other hydrogen. The van der Waals interaction is the small attraction that is created from this slight temporary separation of charges, shown here by the dotted lines. In the next instant, the electrons switch positions and the hydrogens switch charges, but the weak attraction remains. This process repeats over and over at each carbon pair made by the two chains. Now, let's zoom back out to see both chains. A single pair of attracted hydrogens would be easily broken, but when this type of van der Waals interactions occurs at multiple points along an entire hydrocarbon chain, it creates a significant holding power between the two molecules. Disrupting the number of interactions that can occur between fats increases their ability to separate from each other and increases their fluidity. We will discuss three properties that alter the effect of a fluidity on fats. One is temperature, chain length, and saturation. The increased molecular motion created by increased heat causes the hydrogens in adjacent fatty acids to twist and spin away from each other, breaking the induced dipole interactions that held them together. Because the interaction between two fats are weak, it doesn't take a very high temperature to increase the molecular motion beyond the capacity of the van der Waals interactions to hold them together. And many fats that are solids at room temperature will start to melt at body temperature. The slower movement produced by lower temperatures facilitates van der Waals interactions along the hydrocarbon chain, making them more solid. So as a rule, we can say that increasing temperature increases fluidity. Longer chains, like the ones on top of this view, can make more van der Waals interactions along their length, and that requires greater heat to pull them apart. Shorter chains have less bonds and are easier to pull apart and form liquids. Thus the 12-carbon laurate forms a liquid at 43 degrees Celsius, while its 18-carbon cousin stearate melts at 70 degrees Celsius. So our second rule states that decreasing the chain length increases fluidity. Finally, dealing with saturation, as we learned in a previous video, when a double bond occurs in a carbon chain, there are two possible geometric isomers that can occur. Cis double bonds occur when the carbon chain enters and exits on the same side of the double bond, and trans double bonds occur when the carbon chain enters and exits on opposite sides of the double bond. These bond types can have a big influence on the fluidity of the fatty acid. When it comes to fluidity, saturated fats and trans unsaturated fats tend to form solids at room temperature, while a cis unsaturated fat of the same chain length usually forms oils. The reasoning for this phenomena can be explained by comparing the three structures. Saturated chains tend to form long, straight chains that permit lots of van der Waals interactions between adjacent hydrogen. Trans double bonds look a lot like saturated chains, despite their double bond. They tend to produce long, straight chains with lots of adjacent hydrogens available for interaction with other fats. Cis double bonds, on the other hand, create a kink in the chain that keeps some of the hydrogens from being able to get close enough to form van der Waals bonds with adjacent chains. The lower degree of bonding allows cis fats to be more fluid. This effect is heightened by multiple cis double bonds, making polyunsaturates even more fluid. So, our final rule is cis double bonds increase the fluidity. Phospholipids are a class of lipids that form the fabric of biological membranes. Many of the principles we discuss with fats, such as numbering, saturation, and fluidity, also apply to the phospholipids. We will spend a little time showing the structural differences between phospholipids and lipids, and discuss their role in forming a membrane. Phospholipids have a structure that is very similar to the fats, with the exception that one of the acyl groups on the glycerol backbone is replaced with a phosphate functional group. The phosphate typically carries some kind of polar molecule attachment that is represented here by the letter R. Phospholipids are amphipathic, meaning that they have both a polar and nonpolar region in their structure. The hydrophobic region is created from the nonpolar fatty acid chains of the two acyl groups. These are often referred to as the nonpolar or hydrophobic tails. The glycerol backbone, phosphate, and polar R group form a hydrophilic region that is referred to as the polar or hydrophilic head. This dual nature is critical for the phospholipid's role as the primary fabric of membranes. When phospholipids are mixed in water, the water molecules surrounding their nonpolar tails cannot form as many hydrogen bonds. This gives them a higher energy state and makes them uncomfortable, shown here as frowning red water molecules. When two phospholipid tails associate with each other, the water molecules get pushed out of the way, and they reduce the number of water molecules needed to surround that hydrophobic tail region. This recreates a more comfortable state for all the molecules involved. In fact, it might be said that water molecules will actually push phospholipids together in order to increase their own comfort levels. This process is referred to as hydrophobic collapse. When carried out on a larger scale, the phospholipids arrange themselves in a phospholipid bilayer. The polar heads orient to the outside of either surface, while the nonpolar tails are pushed into the middle, away from water. Here, they are able to form van der Waals interactions that lend additional bonding strength to the bilayer. A lipid bilayer is important because it defines the borders of each cell, as well as organelles inside the cell. The nonpolar tails form a hydrophobic barrier that keeps most soluble cell materials concentrated inside the cytoplasm. This barrier is very effective against things that are large. This means that things like this large purple protein cannot cross the barrier on their own. Polysaccharides and even most sugars cannot cross the barrier on their own either. The barrier is also very effective against anything that has a formal charge, even if it is small. This means that ions like sodium, potassium, calcium, chloride, or phosphate are effectively repelled by the bilayer. Nonpolar molecules like carbon dioxide and oxygen can diffuse across the large hydrophobic region. And water molecules, even though it's polar, can still diffuse through the bilayer because of its small size. Because some things can cross the bilayer on its own, the bilayer is said to be semi-permeable. However, the bigger and more polar you are, the harder it becomes to cross the barrier. Fats and phospholipids both share a glycerol backbone and fatty acid tails. Another group of lipids, called the steroids, are chemically different. We will give a brief overview of this family of lipids by discussing their common chemical structure and by giving various examples that illustrate their function. The steroids are characterized by a set of 4 hydrocarbon rings that are fused together. There are 3 hexameric rings and 1 pentameric ring. There are many different structures for steroids that come from changing the type and location of different functional groups on the ring structure. Here we see the structure of cholesterol, one of the best known steroids. It is primarily identified by the long hydrocarbon chain branching off of its pentameric ring and the single hydroxyl group at the opposite end. Together with phospholipids, cholesterol is an important component of biological membranes. Testosterone is another well-known steroid. Structurally, the fused rings stay pretty much the same, but we have replaced the hydrocarbon with a hydroxyl functional group, and the bottom hydroxyl group was replaced by a carbonyl group. Like most of our other steroids, testosterone isn't found much in the membrane, but it does play an important role as a signal molecule. It is the signal hormone associated with male characteristics in mammals. Progesterone differs from testosterone by converting a single hydroxyl group into a methylated carbonyl group. Progesterone plays a role in regulating pregnancy. Like most other steroids, testosterone and progesterone are cell signal molecules that transfer chemical messages from one cell to another. This is cortisol. It has a number of different functional groups, so we won't bother naming them all. The take-home message is that it still has the same fused ring structures. Cortisol itself is an important stress-regulating hormone that increases glucose in the blood and awareness in the brain. Here is an estradiol. This is another name for estrogen. It functions in cell signal pathways that determine female characteristics. Most steroids are signal molecules that activate cell signal pathways. Several of these pathways involve performance, like cortisol, or gender characteristics like testosterone. In society, the term steroid is often used to describe performance or body-enhancing drugs, and many of the steroids will do this. But in biology, we want to emphasize that the term steroid actually describes the four-fused ring structure that is common to these drugs. At least one advantage of this hydrophobic ring structure is that it allows many of the steroids, like this cortisol, to pass through the plasma membrane without any transport proteins. Once inside the cell, the steroids will bind to an intracellular protein in the cytosol. Binding to cortisol activates the protein and allows it to promote the next step in the pathway. Now that we have presented both the phospholipid and steroid groups of lipids, let us take a moment and review what we have learned. Phospholipids are structural cousins to fats. They have a glycerol backbone, two acyl groups, and a phosphate group. Phospholipids are amphipathic, meaning that they have opposite hydrophobic and hydrophilic regions. Phospholipids spontaneously form lipid bilayers with a thick hydrophobic core that is semi-impermeable to many solutes. Nonpolar and small polar compounds are able to travel across bilayers, but large and charged molecules cannot. The steroids are lipids created from a common four-fused ring structure. They differ in the attachment of functional groups to the ring structure. Most steroids are signal molecules, and they have the ability to cross the bilayer on their own without transporters. Finally, cholesterol, which is also a steroid, is an important component of membranes. We hope you enjoyed this video tutorial and found it useful in learning new biological concepts. This tutorial was completed in May 2014 as part of the Biology Education Animation Research Project at Brigham Young University, Idaho. This video was designed, produced, and narrated by Dr. Stephen Christensen. You can find other similar videos for different classes in the biology department online.